Okay, thanks a lot for inviting me for giving this talk. So this will be on the work that was published last year and done probably a couple of years back. It's on time-dissolved crystallography on photosystems using X-ray laser. So I'll try to make this talk as much as possible, not redundant with information that we gave, and try to make it more specific to photosystems. But if there is at all any redundancy, please bear with me. So why the whole fuss? This is the Cox cycle right at the center of the bubble, and it basically represents the catalytic activity of the oxygen evolving cluster, which is a part of the huge photosystem 2 molecule biological system. And some of the most important consequences if we end up solving or identifying and characterizing the entire catalytic process would be, first, it would give us a breakthrough in terms of pushing towards artificial photosynthesis, as you can see on the bottom right. And that is important because whether you do, whether it's natural or artificial, the two important byproducts of such a process is that one, you generate oxygen, and we all know why it's important. And the other is it reduces or it consumes carbon dioxide, which is great again because you're reducing the amount of greenhouse gases that are afloat. And the most important thing is if you consider this as a reaction, a chemical reactor, then your inputs as fuel is water and sunlight, which is perennial, it's renewable, and it's non-polluting, which is great. So it's almost like an ideal reactor. So there are multiple excited states that the oxygen evolving complex, which is manganese 4, calcium, oxygen 5. That's the elemental system that compromises the cluster. There are multiple excited states that accompany each of this catalytic cycle, the catalytic stages in the cycle. And conventionally, you take S1 to be the ground state. It's called dark, which has been marked with black. And then with each subsequent photoexcitation, you achieve the higher excited states, S2, S3, S4, S0, etc. Now, for the case of this work, they focus predominantly on the second excited state, which is S3. So for all future references, I would either call it S3 or the 2-flash system or 2-flash state. And as a comparison, we'd call the ground state as the dark state or the S1 state. So the idea is to try and characterize as much as possible what is different or similar about the state 3 in the catalytic cycle as compared to state 1. And here and there, I'll take some reference to some of the experiments that were done a little later on characterizing, if possible, the S4 state to just bring in comparison and make better understanding out of it. So the way I'll approach this talk is first to give a brief introduction on the multiple aspects of modern experimentation, which is sample growth and characterization, because you need to know how to grow your samples and do the basic characterizations to make sure that it's worth going for an experiment with it. And then sample injection. More and more complicated systems of experimental setups nowadays require very different and innovative techniques to help the sample interact with your probes. So we'll learn about that. And the third is the experimental setup in itself. So the LCLS is, well, for us, it's very, we know it well, but in general, it's a very new technique of laser component probe, in this case, because we're doing time resolved. So we'll talk about what is the experimental setup and what are the parameters going with the experiments, this specific experiment. And the last, we'll focus on data analysis, because most of the data nowadays that we get with such high efficiency and highly quantitative work that we are aiming to do requires a lot of post-processing of the raw data that we get. So we'll focus on each of these one after the other, and finally we'll look at, in the specific case of photosystems, what is the science that we unravel, or what are the observations we make. So first with sample growth. So typically protein crystals are grown in batches using various very common techniques. People would be familiar with diffusion or hanging drop methods. But the problem with photosystems that people have seen is that they yield a very broad distribution of crystal sizes and typically large. And they also have a lot of defects. So in the case of using the SFX at LCLS, where you're looking at typically microcrystals, roughly of the order of 5 microns or lesser, you want to control the size and also get a very homogeneous distribution of your crystals, which are pretty much around the same size. So what the chemistry group at ASU, headed by Petra Fromm, did is to use a free interface diffusion technique, which is a very simple technique if you think of it, and very smart, which is that you have a two-phase system. One is the protein and another is the precipitant. The precipitant is typically at a higher density than the protein, and then you have the nucleation happening right at the interface. The cool thing about this is that as the crystals grow, as they nucleate and they start growing into larger sizes, obviously because they accumulate more mass, they're going to settle down. And because the protein and the precipitant form a two-phase system that don't mix, the idea is that as the crystals start becoming dense and going down, they stop growing. So it's a natural inhibition, and it helps in controlling the sizes. So the only thing that you need to work around is the optimal concentrations and what constitutes the precipitant and the protein solution. So once you do that, you can obtain pretty good crystals at very good homogeneous densities. And the way you check whether you have good crystals is by characterizing them using a various number of techniques. What you see on the left is the standard crystals that people have grown so far for synchrotron, which is huge, roughly around 300, 500 microns at the top left in figure B. And then what you see on the right, top right, figure C, is, for example, an optical image of a batch of Photosystem 2 crystals grown at ASU, which are roughly about 1 to 2 microns in size. And the way you characterize the size, if you're not able to get such good accuracies with an optical microscope, is typically by using either DLS or sonic, or like both of them together. So the bottom left figure shows you a size distribution of a batch of PS2 crystals grown, and you can see that it's pretty much centered around the 1 micron radius. Whereas on the bottom right is sonic. So sonic is an important instrument in the sense that biocrystals are typically supposed to be non-synthosymmetric, and it helps in identifying whether the crystals that we have are good crystalline materials, or do they have like bad crystals, defects, or are they amorphous and they don't make good crystals in spite of the size distribution. Because when you put them through sonic and you try to get an image of it, they only image well because of the second order nonlinearity. They only image well when you have like a crystalline sample and that does not have a center of symmetry. So what you see at the bottom right is a demonstration of that. The other thing is also that sonic can go much further than DLS in identifying the size distribution. So if you ever had crystals that were not going to 1 micron or 5 microns and have like a few hundred nanometers, you'd probably be still able to identify the distribution using sonic. Now, once you have had the right crystals, the next question is how do you deliver them, or how do you help them interact with your instrument? So typically that's done in the LCLS using a liquid jet. There are multiple variations that have eventually spun off, but the idea is basically this, that when you are interacting a very intense 10 to the 12 photons in a 50 femtosecond pulse, you're basically going to destroy the material. So this is not, it's not a soft technique, it's an invasive technique. It destroys every bit of sample or solution that is around. So the idea is, can we flow a jet, and can we control the speed, and can we control the flow in such a way that it maximizes the interaction of the jet with the X-ray beam. And here's a beautiful, a small video showing what actually happens. I'll let you watch the video and then describe it. If I get the cursor. There. So what you see in blue is the actual liquid jet, and what you see in white is the gas focusing of it. So traditionally, if you think of a liquid jet, it's basically like water flowing out of a tap. But what's different here is that you're trying to make a small beam. The reason why you want to make a small liquid jet beam is because the crystals are about 1 or 2 microns in size, in diameter. So if you have a very huge diameter of liquid jet, like 50 microns, then you're going to get a majority of the scatterings from the solution. And it's the probability of hitting the sample also goes down. So you want the liquid jet diameter to be comparable or slightly above the size of the crystals. So the way you do that is by using gas to focus the liquid. So let me just play through that animation once again. And as you can see, the liquid jet is compressed to a size that is substantially even smaller than the nozzle tip. And then eventually it flows out with the crystals, where the red beam that you see is the LCLS XFEL hard X-ray laser beam interacting with your sample. So every time it hits, so you're going to obtain some scattering pattern on your detector, and that's how you hope to make sense of the experiments that you're performing. So now that we know how the GAVL works, let's go straight to the experiment. So as explained in the previous talk, it's going to be another time-resolved experiment setup. where you have a pump laser and a probe laser. The actual geometry looks somewhat like this, where you have all the liquid jet flowing in one of the axes. You can label that as Z, vertical, going down, and then the X-ray laser is moving in one direction, and the visible laser is orthogonal to it. And the time scales of the triggering, the pulse widths, the pulse durations, the interflash timings are all presented at the bottom of the screen. They are generally calculated assuming kind of having an idea of what is the reaction half time for the products to form, so that you take 3 times the half time. That gives you about 90% of product formation.
Could you please talk?
So those are most of the parameters. I'm not going to go into the details of it, but just want to let you know that the diameter for the liquid jet was about 4 microns. And the most important thing is, because these are photoactive, which is why we're using a visible pump, we generally coat the nozzle black, except very close to the tips, which is where the first flash typically excites into the first excited state. But going on, so once you do all this, the whole idea is to get some scattering onto the detector and hopefully make some meaning out of it. So what all is involved in that process, we look under data analysis. So in the data analysis, you have multiple steps. The first thing you do is start with pre-processing, which is clear out the unnecessary noise. One of them is dark currents, which is present at all times. You want to remove that. You want to take account of that. The other is dead or saturated pixels. The intensity is so high that sometimes some of the pixels on the detector can get saturated. So you also want to account for that. And typically, you assume that every interaction of the XFEL pulse is going to produce some scattered pattern on the detector. Well, hopefully some. Now, whether it's useful or not is characterized by whether or not, for example, in this case, we have 25 or more significant peaks, diffraction peaks, or intensities on the detector. And the way it is characterized is the analog to digital unit conversion of a factor of about 400 or higher. And because this is a time-resolved experiment where we want to compare the ground state, which is dark, and the double flash excited state, you can easily separate those because the experiments go alternating in scheme, as is mentioned here. As is mentioned here, where one of the pulses happens at 2 times the frequency, which is the X-ray pulse, and the pump pulse happens as half the frequency. So every alternate pattern that we get is from the excited state. Well, there are auto-indexing suits available. MOZZFilm is one of them. In the case of PS2, it's expected to be orthorhombic, so you start with that assumption. Now, in this case, we see that there is a difference in the unit cell parameters for the dark state as opposed to the excited state. I'll discuss as to how it is significant a little later, but you can identify, the whole point of mentioning that here is you can identify that as and when you start indexing. And what we also see is that when we do the 3 flash experiments going to the S4 state, hopefully to the S4 state, we identify that the unit cell parameters, sort of revert to what we see in the dark state. And again, I'll justify that in just a moment. So the next step that you do is you have to merge all these intensities. So yes, you have diffracted intensities, but you need to make meaning out of it. So what you try to do is you first use a multicolor type of merging, and it's essential that you have multiple such data patterns to merge from, because most of the intensities that we get are partial reflections, which is that the evolved sphere does not cut through the entire volume of a diffraction in its 3 dimensions. So you want to have as many intensities collected from the same diffraction spot, and that like that of every other diffraction spot, to account for its ideal form. And once you have that, you can go ahead and obtain the structure factors, assuming a kinematic diffraction theory. And then typically at this point, you can do certain internal consistency checking by, for example, aspect where you divide the data set into 2 halves and try to see whether they still give consistent data within a particular amount of error. And once you do that, you go to the actual structure determination, where you use these intensities, couple them with phases that are obtained from existing models, and then try to make meaning out of it. For example, in this case, we start with molecular replacement. The PDB that's used for that is mentioned here, CHARC. water, lipids, and other detergents have been removed, and then the monomer 1 on the PDB file has been used for search. Then once you do that, you can go ahead and do some refinement processes. In this case, they're not doing rotational refinement, they're doing only translational refinement. And because we find that when we're doing some of the preliminary analysis, we can get a good idea of what the resolution of the overall diffraction pattern is going to be, or the overall electron densities when we plot going to be. And so because it's not as high, it's about 5 angstroms, so the B-factor refinement has not been carried out because it's not as relevant. And of course, at this stage again, you can calculate the R work and R free, which are again statistical parameters that tell you how good or how ideal your refinement procedure is. Then coming to the actual electron density. So once you have the phase information, you can plot the electron density as was shown in the previous talk. But there are various other ways of making sure that the model that we use does not impose a huge bias on the information that we get when we just put in the diffracted intensities and get out the electron densities. So one of the ways to do that is by calculating an omit map. An omit map basically is omitting a portion, certain residues, or in this case, for example, the oxygen evolving cluster, from the actual refinement procedure, and then overlapping it with, or finding other ways of difference map or something like that, where you can compare the non-omitted version with the omitted version and try to see whether there is a bias that is arising out of the model or not. And another way of doing it is also simulated annealing, which is you basically heat up the molecule virtually to a very high temperature and let it cool down, and try to see whether, as it cools down, does it get back to the atoms and to the molecules bonds, do they get back to similar positions? And from there you can figure out again whether there is a bias from the payload of the models. Now, having known what we generally do with the data, let us come to the actual science of what happens with the PS2 when they did the experiments. So the first thing, as I mentioned earlier, is that when they do the indexing and then refinement of the index patterns, they're able to notice a lattice parameter increase. If you can see, you would probably want to look at the data row-wise. The first row is the dark state, the second row is the double flash S3 state, the third row is again dark state, which was done as a separate experiment to go to the S4 state, which is the last row, the third 3-flash state. And what you can see is that in the second row, especially at along the A-axis, which is the first plot to the left, you can see that comparing the dark state and the 2-flash state, there's a clear 3 to 3.5 angstrom increase in the unit cell parameter. Now, the resolution is not high to pinpoint what the exact cause is, but the idea is that because the plastoquinones at this point obtain 2 electrons and they are free to move away, one of the plastoquinones is free to move away, it's possible that the movement of the plastoquinones away from the crystal sites that they were actually supposed to be in, because they form a crystal contact region, would actually reason out to be the cause of the expansion in the unit cell parameters. And an indirect way of confirming this is done in the latter half of the figure, where they show the 3-flash system, which seems to, if you notice, it seems to resemble what you see in the dark state. And that shows that when the plastoquinone derivative was added for the 3-flash experiments, and the plastoquinone was present, so it was not that the plastoquinone was not present after the second flash, you notice that it is able to revert back the S1 state. So it kind of gives a leaning towards the observation that we think here is because of the movement of the plastoquinones. And the second big change that we see here is a change in the structure of around the oxygen evolving complex. Now, the top graph, the figure on the top is the dark state, and the figure on the bottom is the one with the 2 flashes. And what you see is that as compared to the dark state, in the excited state, in S3, the cubane, which is removing the, if you leave out the dangling manganese, which is one of the 4 manganese, the rest of the system, Mn3OXCA, seems to shrink closer. And the distance between the dangling manganese and the oxygen evolving complex seems to have increased. Now, there have been various theoretical studies, various spectroscopic studies that kind of lean towards this idea. The idea is that the angular distortion that you expect in some of the lower S states, because of the manganese valence being more around +3 and not around +4, is absent when you go to some of the higher S states like S3. And what this does is the distortion that forces the bonds to move away and to clear the steric hindrance and take a longer bond length is absent when you go to S3, and so the bonds are much shorter. What you also see is outside of the oxygen evolving cluster, the transmembrane helices, some of the loops seem to relocate themselves. For example, as you can see in the dark state simulated annealing omit map at the bottom, you can see that the loop AB with D61 is seen to move closer to the cluster, whereas the loop CD with D170 seems to move away. Now, there was an interesting studies done some time back on whether the activity of D170, where they mutated D170 and they tried to observe whether that has any effect on the oxygen evolving evolution as such and the catalytic activity of the cluster. They didn't see anything. And what is interesting is that we as well don't see any change due to the movement of the CD loop away from the oxygen evolving cluster. Now, so for example, this could be a first order confirmation of the mutagenesis studies that were done earlier on the PS2 systems using FTIR spectroscopy. So right. So to briefly conclude as to what we have, what the work has aimed to do and what the work has achieved, it was basically a first proof of principle of a successful time-resolved serial femtosecond crystallography done at NCLS on a biologically relevant system as PS2. Of course, the S1 and S3 states were probed, and the visible form was used with a double flash scheme to excite it to the higher excited states. Now, there are two important scientific relevance that came out of this experiment. One of them is the variation in the lattice parameter, which is that we see the unit cell parameters increase when we go to the excited state S3, and we believe it is probably because of the dissociation of the plastoquinones and that affecting the the cells being together, the cell contact. And the second important thing is the structural variation about the oxygen evolving cluster. As mentioned here, the cubane shrinks when we go to the S3 state, and this is probably explained by the prior theoretical studies and experimental studies of Yantarder type distortions. And what is interesting is that it is also possible to host as a ligand the second substrate of water that is required to complete the reaction. Now, also the transmembrane loops, which are just outside of the OEC, relocate. And as mentioned earlier, the AB loop gets closer and CD loop moves further away from the cluster. And as was explained in the previous slide, this could probably be a consistent explanation as observed earlier on the mutagenesis studies on D170. Well, thanks for the talk, sorry for the delay, and I'm open for any questions if people are interested.
All right, thank you, Ganesh. Do we have any questions?
We have a question from here.
Yes?
Okay, so the question was, what is the reason for the absence of the Yantarder distortion? So it depends. It basically depends on the spin states and the molecular orbitals, but it's expected that when you go to S3, all of the manganese go to +4. I can show you. So in the S3 state, all of the manganese are expected to be in the +4. And the +4 configuration has enough space to accommodate all the ligands without a necessity for, well, there's no steric hindrance. That's basically what it's saying. So the bonds are convenient as they are. And the dangling element moves away, so it gives more space for the other ligands to attach closer to them.
And so the S3 states or in the lower states or
Yeah, I don't know if it's clear.
Okay. All right.
So it basically starts with S0, where a majority of them are +3, and then with every single flash, you're increasing the oxidation state of one manganese atom that compensates for the one electron that goes in the cycle. Right?
Okay.
Yeah. Any other question?
So I have a question. My question is, what is the justification of the different Fourier method when the lattice parameters are different between the dark and the light states? I mean,
Right.
is this still justified? I don't know.
So, I mean, of course, the resolution that we have from our data is about 5 angstroms. So, I mean, we can't justify it firsthand, but the idea with which we feel we are consistent in our argument is that the plastoquinones form an important contact point when they form crystals of PS2. So at S3, if you can see this diagram, right around S3, when the S3 state is reached, one of the plastoquinones can get 2 electrons now to move away. Now, when this happens, you're removing plastoquinones out of the contact site, and that can either, it will basically alter the crystal structure, because that is one of the important contact points when they form crystals of PS2. So that is why it is assumed that maybe that is the reason for the huge, variation in the crystal lattice units and parameters.
Any other questions?
No?
Anyone left?
All right. Well, thank you to both of our presenters.